Hello students, here we are once again to examine lesson 2 of the 201 Directives 1 with Maureen Triple, the facilitator. In our last lesson, we looked at Mendel's first law of inheritance. Today, we'll be examining Mendel's second law of inheritance, which is the law of independence assortment. As we go on, enjoy the lesson. In our lesson today, there are some terms we need to know concerning Mendel's second law of inheritance. First is a dihybrid cross. A dihybrid cross is a cross between two parents that differ in two parents alleles. Second is a hybrid. A hybrid is an individual that is heterozygote for two parents alleles. Incomplete dominance. An incomplete dominance is a situation where a dominant allele cannot completely match the recessive allele. Codominance, on the other hand, is a situation where no allele is able to match the other allele. The purpose of the dihybrid cross was to determine if any relationship existed between different allele pairs. Mendel's law of independent assortment, which is the second law, says that the segregation of the factors or genes representing two traits is independent of each other. The law of independent assortment takes place as maternal and paternal sources of DNA are randomly divided during meiosis. The gametes either inherit the maternal version of a gene or the paternal version. Which version it gets is completely random based on the order that these genes lined up during the first stage of meiosis. The underlying mechanism of the law of independent assortment is that genes are located in pairs of homologous chromosomes. Homologous chromosomes have these genes in the same locations on each of them. Though the homologous chromosomes are very similar, they are not identical due to the presence of the different genes alleles on them. During metaphase 1 of meiosis 1, the homologous chromosomes line up at the center of the cell and their orientation is random. This forms the basis of independent assortment. This is what happens during the meiosis. You see homologous chromosomes lying in two ways at the metaphase 1 of meiosis 1. Here they lie in different ways, two different ways, even at the central, the equator of the cell. Then on segregation, here we now have, during gamete formation, we have four different gametes being from two chromosomes. Now we'll examine Mendel's experiment on the law of independent assortment. Mendel carried out some dihybrid crosses on the law of independent assortment. The first we consider the type and color of flowers. A pure-breeding tall plant with red flowers was crossed with a pure-breeding black grass plant with white flowers, as shown by the alleles shown. The first generation of the plant observed, they are all tall and with white flowers. Mendel continued his experiment and went further to cross-pollinate the F1 hybrid plant, and it was discovered that for each of these hybrids, four possible genotypes were formed, as shown there. And at the end of the day, the progenies of this cross, that's the F2 progeny, came out as 9 tall red color flower plants, 3 tall white color flower plants, 3 dwarf white color flower plants, and a single dwarf white colored flower plant in the ratio of 9 to 3 to 3 to 1. That's the hybrid phenotypic genotype. In another dihybrid experiment, Mendel looked at seed color and seed shape. in this experiment is for red, yellow, brown seeded pea and a green wrinkled seeded pea. In that experiment, the F1 generation, they were all yellow and brown seeded pea. Because yellow and brown are dominant over wrinkle and green. First, F1 generation are thought to heterozygous yellow and brown. He went further to self-pollinate the F1 generation to result in set of F2 generation where 9 of them were yellow and round, 3 of them were yellow and green, 3 of them were wrinkled and round, and only one was green and wrinkled in the same ratio of 9 to 3 to 3 to 1 as seen in the former experiment. So most of inheritance, however, do not exhibit the regular Mendelian segregation pattern. This can be seen in the case of incomplete dominance, as seen in a cross of a red flowered snapdragon plant and a white flowered snapdragon plant. As shown in the figure here, the F1 generation of the plant, they all produce pink flowers. Both colors blended, red and white blended to get pink. Hence, none of the colors is completely dominant over the other. Not marked. We said that it's that of incomplete dominance. Why, however, the F1 generation of pink flower plants were allowed to self-pollinate themselves? The F2 generation exhibited all the colors, where we have 1 red to 2 pink to 1 white in that ratio. In other words, 1/4 of the offspring has red color, half has 3 pink colors, and 1 white color. This is in contrast to what was obtainable in the Mendel F2 generation plant. In conclusion, we've been able to look at the mechanism and the basis for independent assessment and examine some Mendel's dihybrid. On the basis of the given task, you should be able to determine whether the pattern of inheritance of the given trait is in line with the regular Mendelian segregation pattern or not. Interesting indeed. Exercise to take home. In a cross between a pure-breeding yellow brown stage and a pure-breeding green wrinkled stage, show the progeny up to second generation, choose single for the seed color and seed shape. In our next lesson, we'll examine the laws of probability. Bye for now. See you in the next class.